Now I'm happy to introduce our speaker, Sarah Schaack, Professor of Biology. Sarah is a first-generation college graduate and went to a small liberal arts college for undergrad. She started at Reed in 2011 after completing 2 postdocs, following the completion of her PhD in biology with a minor in genetics and an MS in zoology with a minor in African studies. She is an evolutionary genomicist studying the dynamics, causes, and consequences of mutation using a combination of experimental, comparative, and bioinformatics approaches. She always endeavors to serve as a champion and advocate for others who are dipping their toe or diving headfirst into the pond of higher education and science research for the first time. Sarah will give a lecture entitled "The Myth of the Mutation Rate: How Recent Discoveries in Genetics Illuminate the Most Important Parameter in Biology." And with that, I'll turn it over to Sarah. Thank you.
Thank you, Shanti. Hi, everyone. I'm so delighted to see so many people here today. Thank you for coming. I'm going to go ahead and share my screen because I did put together some slides, and then my face will appear in the lower corner of your screen on top of the slides. And I just want to say 2 things at the beginning. One is I decided to take a little bit of a broader approach to the lecture today and talk not just about mutation rate stuff, but about mutation more generally, since that is the main question in my lab, but we actually have several spokes to the wheel of mutation research that we do. But if at any point there's a clarifying question that I can answer very quickly, if you want to, you can go ahead and put that in the chat. I won't monitor the chat like super religiously, but I don't want anyone to miss anything because of some small technicality. And then we can talk more broadly about implications or whatever questions you have at the end. So let me go ahead and share some slides. So can folks see and hear me all right? If you can give a thumbs up.
Yes.
I can see you.
Yeah, it looks good, Sarah.
Okay, so I want to start off by saying that I'll be talking about some specific data and inferences today, most of which was collaborative with the folks on this slide. And I've put some notations next to names. The folks who have asterisks next to their names are Reed students, and the folks that are underlined are postdocs that were in my lab at some point in the past. And so I really want to emphasize that none of this was a solo effort, and none of it would have been possible without these other people. So just to make sure we're all on the same page starting off, I want to go over what is a mutation. I think the word mutation has kind of a negative connotation in the popular press. And for me, it does not. I think mutations are the best thing in the whole world. That's why I've dedicated my entire career to studying them. So while you might be thinking of a Marvel Comics type version of mutations, and we will actually get to something like the Marvel Comics version of mutation by the end, mutations generally refer to any of the changes that happen to DNA either during DNA replication. So when the DNA is being replicated before a cell divides, there are a few mistakes that are usually made and not repaired, or changes to DNA because of damage from the environment. So think of things like UV, X-rays, chemical carcinogens, etc. So the way that these mutations manifest is in a couple of different ways, and I've illustrated a few of the main ones here. Base substitutions are simply, that's the technical term for changing an A to a T or an A to a C or an A to a G when you think about the letters that make up DNA. There's bigger mutations like insertions of a chunk of DNA, a stretch of letters, or deletions of a stretch of letters, and those are broadly referred to as structural variation or structural variants. And there's also larger scale mutations caused by pieces of mobile DNA. So mobile DNA is a class of DNA that actually can encode for proteins that facilitate their transposition around the genome. And that means replicating and inserting somewhere in the genome or excising from a spot in the genome. There's actually a lot of different acrobatics that mobile DNA can do. And even though and these are just some examples, there's other categories of mutation as well. Even though there's all this diversity to mutations, almost everyone who studies the mutation rate restricts their study to that first class of base substitutions. And that's not because those are the most common mutations or because those are the most important mutations. It's because those are the easiest mutations to study. Another fine point, I would say, in mutation biology, no, it is not always A that gets substituted. That's just an example, is that we usually think about mutations as happening perhaps between parents and their offspring. Those would be mutations that are inherited, but mutations, of course, can happen in an individual, and they just happen in the cells of that multicellular individual. Those are somatic mutations, so they're not heritable. They will not be passed on to the next generation. And there's also a distinction to be made between mutations that happen within a cell because of damage or errors inherent to that cell, and mutations that happen because of the receipt of DNA from another lineage. And horizontal transfer is the broad name for that type of mutation, when the change to the DNA is actually the insertion of DNA from another lineage. We use a special term for that type of mutation, and those are horizontally transferred pieces of DNA. And that is the Peter Parker getting bit by the spider and turning into Spider-Man type of mutation, which you might think of as being just the stuff of comic books, but actually is prevalent in both unicellular and multicellular life. So my lab has been focused on two parameters in biology, estimating two parameters. And one of those is the rate of mutation, which is represented by the Greek letter mu. And one of those is the phenotypic effect of mutation. What does the mutation do for the organism, which is called alpha? And I use mu and alpha not to be fancy or because I want you to have all these equations in your mind between phrases and symbols. I use it because it saves me a lot of room on the slides. So I want you to have mu certainly in your quiver. I'm going to save that question for later because that'll get us a little bit off track. Oh, can you see the bottom of my slide even though I can't see the bottom of my slide? Okay, great. In every case, if I have a kind of a take-home message and I want to help everyone get back on the train, if for some reason you thought about something else for a minute, that take-home message is going to be in red at the bottom of the slide. So estimating those two parameters, mu and alpha, what is the rate at which mutations occur and what is the effect of mutations are really the base questions for a bigger, more beautiful question in biology, which is how do mutation rates evolve? And many people just think of mutation as being this fact, maybe even this unavoidable fact, but the reality is that the mutation rate is a trait, just like the average height of individuals is a trait, or just like the coloration, the pigmentation in feathers is a trait, or flowering time is a trait. The rate of mutation is a trait. And so that's how we study the mutation rate in this very matter-of-fact way. We want to know what the mean is of this trait and what the variance is of this trait for a species. And the reason we want to know that is because that parameter mu is critical for so many other questions in biology. Why is it critical? It is critical because mutation is the ultimate source of all genetic variation. So if we don't have mutation, we don't have genetic variation. And if we don't have genetic variation, we don't have evolution. And if we don't have evolution, we are all just fragments of DNA still floating in the primordial ooze. Let me see this question. I didn't see it before it went away. It's not a meta trait, but it is a complex trait. So it's a trait that has many inputs. And it's true that the mutation rate, you would want to be more specific. Are you talking about the mutation rate when you're talking about base substitution rate, or microsatellite mutation rate, or insertion rate, or deletion rate? And we'll get to that soon. Can the distribution be multimodal? In theory, but we have typically seen that it's like most continuous traits, it's not necessarily normally distributed, but Poisson distributed. Okay, why else do we do this work? We like to do it because it's hard, and doing things that are hard is more fun than doing things that are easy. And the reason why mutation rate estimation is hard is mainly because the rates are so low. We're talking about 1 change per billion base pairs per generation, or maybe even less than that, maybe 1 change per trillion pairs per generation. So it's hard to observe these events, and thus it is hard to calculate a rate, but it is possible to do. And we like to do those experiments. They're multi-year experiments, even on the small microcrustacean that we work on. It would be very hard to directly estimate mutation rates in long-lived organisms, although it is possible to do that. It just takes tons and tons of money. All right. So why do we care about all of this? I mentioned that it's a critical parameter in biology. Some of the paradoxes in biology that could be addressed by knowing the rate of mutation are listed for you here. So when we started to sequence things, the data right away indicated that there was way more genetic variation among individuals and populations than one would expect if natural selection was indeed getting rid of any variant that was not favored in the environment. So this is a big, it's called Lewontin's paradox, or the paradox of diversity, or the diversity paradox. And the big question in biology since sequencing has started is, why is there so much genetic variation if natural selection is getting rid of most of it and mutation rates are low? Another big question is, why is variation not correlated well with population size? So the more individuals in the population, the more targets there are for mutation, the more mutations you would see. But in fact, there's not a super strong correlation there. Does crossing over count as mutation? That's a very borderline case. Crossing over can induce mutations where the double-stranded repair happens, but most people don't think of it as a mutation. I think of it kind of in the portfolio of mutation-causing mechanisms. There are also lots of practical reasons to care about mutation rate. This is how we predict disease prevalence in populations, or the risk that folks have for genetic diseases if they were to mate randomly in a population or not so randomly. It's very prevalent right now in our understanding of SARS-CoV-2, the evolution of variants that are affecting our everyday lives. It's relevant for aging. There's a lot of interest in somatic mutation rates related to aging, and cancer risk. A whole slew of cancers are genetic in their origin. And if we know more about the kind of damage that occurs that causes cancer, or the rate at which spontaneous mutations occur that cause cancer, we have a better chance of targeting which cancers we should study and try to cure. It's also really relevant for conservation biology, for the kind of charismatic megafauna that you might be interested in preserving, you and others, as well as for people interested in de-extinction efforts that involve the precise hybridization or genetic engineering of things like bringing back the woolly mammoth. So understanding mutation rates is important for many reasons. I hope I've convinced you that it's worth listening to the rest of this talk. So I am not the first person to study the evolution of the mutation rate by a long shot. And in fact, many people have studied it for a long time, mostly looking at rates across species. So what do I mean by that? I mean, we usually have at best a mutation rate estimate for 1 or 2 individuals, and we hope that that represents the mutation rate for that entire species. And so you'll often in mutation rate papers see plots like this, where every single individual dot on this plot is the average estimate for a species but it's usually based on only one individual. And people will try to draw broad conclusions about how mutation rates must evolve because they'll see in this example a relatively robust looking correlation between that mutation rate, which is plotted on the Y, and some attribute that they think explains differences in mutation rate. Here on the X, that attribute is the effective population size, the number of individuals in the population that are able to contribute genetic material to the next generation. I won't go into showing you the data in support of or that are not consistent with each of these theories. But I do want to make one important point here, which is that all of the theories that have been developed to explain how the mutation rate evolves predict low variation within a species. They all rest on this idea that mutation rates are higher in some organisms and lower in other organisms, and we don't expect there to be a lot of variation within a species. We expect most of the variation to be between species, such as these dots of species that I'm showing you here that are then color-coded by kingdom or domain in the tree of life. So what does my lab do? We ask that question, the question that everyone has overlooked for 40 years of mutation rate evolution research, looking at intra-specific variation within a species. How much variation do we see when we actually measure the mutation rate for more than one individual from a species? And so in this plot, I'm showing you the sum total of data, which is on the base substitution rates. So that one kind of very typical form of mutation where you're substituting a letter for another letter, though it's not always A, for species where we have done the experiment. So this is direct estimates of the mutation rate for all the animals, I should say, not all species, all the animals that we have done these experiments. And you'll probably first note, we have not done these experiments for very many animals. There are 3 species of worm here, Caenorhabditis elegans, which is shown for you here, Caenorhabditis briggsii, which is nearly identical phenotypically, and then Pristionchus pacificus, another roundworm, which is almost 200 million years diverged from these 2 Caenorhabditid worms, but still looks almost identical, are 3 of the 4 species for which we have these direct estimates in animals. The 4th, of course, being the genetics workhorse, Drosophila melanogaster, the fruit fly. And until our lab started doing these kinds of experiments, this was it for animals in terms of the empirical estimates. So what did we find? We looked at another species entirely. Doesn't one have to consider DNA repair mechanisms? So these estimates are whatever are the number of mistakes or damaged pieces of DNA that go unrepaired. So yes, that the variance that we're seeing could be because there's not actually a difference in the input of mutations, but there's a difference in the effectiveness of DNA repair. But we can't do in any of these organisms the type of what I would call pre-mutation rate estimate testing. So what is the mutation rate that then DNA repair repairs? You can do that in yeast, but it's very, very tricky to do that kind of experiment. So this is the net mutation rate given endogenous DNA repair that's happening. These are all great questions, by the way, no surprise. So we conducted these same kinds of experiments. They're called mutation accumulation experiments. And the gist of a mutation accumulation experiment is that you take an individual, a single individual, in this case, we're going to do this with Daphnia, and you let her have 100 daughters, and then you propagate those 100 daughters separately in a lineage for 5 or 10 generations, every generation propagating the line with a single randomly chosen offspring that may or may not have some mutations that have occurred. And at the end of 5 or 10 or however many generations you can bear to wait, you sequence the descendants. You also sequence the grandmother of the whole experiment. And then you literally count how many mutations happened in the descendants after 10 generations relative to the grandma that you also sequenced. And you divide the number of changes that you see by the amount of time or the number of generations that have elapsed. So it's a very not rocket science experiment to do, but it does take a lot of time. And I am glossing over how hard it is to actually observe many of the kinds of mutations beyond those base substitutions that I showed you an example of at the beginning. So why do we do this work in Daphnia when everything else that it's been done in are even more common classical genetic model systems? Well, I've been working on Daphnia since 1996. So part of it is just a historical inertia that I like Daphnia, and I've been working on them for a long time. But part of it is because in ecology, ecotoxicology, and in evolutionary biology to some degree, Daphnia are one of the most well-studied organisms out there. So they are very key parts of most of the early work on predator-prey dynamics, community ecology dynamics. They are very much a bioindicator species. So if you're worried about the chemicals in your water, one of the first things you're going to do is find out what the level of that chemical is will kill Daphnia. If Daphnia populations collapse, fish populations collapse right afterwards. So they are an important model organism in many other subdisciplines of biology, but they have not been a workhorse in genetics until relatively recently. Oops. All right. In addition to the relevance that they have in biology more broadly, they also have this very unique reproductive cycle, which is they can reproduce. Can you see my cursor, Shanti? When I
No.
Okay, so in the basically the middle of the slide where it says ameiotic parthenogenesis, they can reproduce asexually. That's a fancy way of saying one of the forms of asexual reproduction. And they can also reproduce sexually. And so in our studies, sex is actually a nuisance variable in terms of studying mutation. We don't want the organisms to be able to get rid of mutations that might have occurred by producing haploid gametes that only have a single copy of their DNA, because that's the major. Sex is a trait that has evolved in order to facilitate the purging of mutations. So in our experiments, we're able to harness the fact that Daphnia can reproduce asexually and eliminate that nuisance variable unless we want to study it. So what did we find when we did mutation accumulation experiments in Daphnia relative to these other species for which we had some estimates of the mutation rate? And I hope that you'll notice that in most of these cases, the estimates for the mitochondrial mutation rate, which are shown in with the triangles, are almost the same across these 4 taxa. And then the estimates of the nuclear mutation rate are 2 orders of magnitude slower. And they're also very similar across all 4 of these taxa. So the lack of interest in intraspecific variation prior to our work, I have to say, was relatively justified, because from the data that we had, there didn't look to be a lot of variation within or between species, as long as you were looking at tiny invertebrates. So what did we see with Daphnia? Well, I'm showing you the data from 2 different species here. One is Daphnia pulex, which I worked on when I was a grad student and a postdoc. And one is Daphnia magna, which is the system that I use in my lab and I have for now 10 years. And if you look at the nuclear rates in Daphnia pulex and Daphnia magna, you'll see that on average, they're higher than any of the rates that had been reported previously. And what's really noticeable is that the mitochondrial mutation rates are extremely high in Daphnia. What does this mean? We're not sure, but it definitely signals to us that more people need to be looking at more taxa in order to measure these mutation rates, so we can start to say things broadly and confidently about the evolution of the mutation rate across species, because our data really broke the mold that had been established. Vertical bars are when they set up replicates. So that is a confidence interval. So is this important? Is this interesting? Well, the take-home message of that section, and this is the only vignette of our research that I'm going to show you, lots of graphs of data where you really have to look at the symbols. We'll transition to some more broad questions here. Let's see what my time is. For our last 5 to 10 minutes. But the amount of variation that we saw is represented in this far right column by orders of magnitude of variation. So we actually measured mutation rates for many different categories of mutation, which are listed in each row here, not just the base substitutions that most people focus on. And that was a novel contribution of our lab. But on top of that, doing this intraspecific comparison and showing how many orders of magnitude of variation really challenges all of the prevailing theory aimed at explaining how mutation rates evolve, because the variation we saw within one species was larger than the variation people were seeing across a kingdom. So the type of mutation that I mentioned to you at the very beginning, those caused by mobile DNA, is actually my favorite. This is my passion. I'm not that excited about base substitutions, but I do have to measure them because that's the currency of mutation biology that most people think in. But transposable elements and their cousins, endogenous viruses, are basically the two main categories of mobile DNA. Yes, it would be great if someone did this in echinoderms. It would be great if someone did this in pretty much anything. But I'm going to transition from mutation rates now, and then we can come back to that during the Q&A. The types of mutations, as I mentioned, that might be not only more frequent, but also more impact, are these larger scale mutations where you're talking about whole pieces of DNA inserting or moving around in the genome. And transposable elements and endogenous viruses are especially interesting to me because when they get into a genome, they can start to jump around and replicate. And they often have in their sequence, you could think of it as a payload. They might have either a protein that they code for, or they could have a snippet of the genome from where they originally jumped from that they bring to a new position. It's not just changing one letter or a random insert of, and not an insert of a random piece of DNA. It's a piece of DNA that's capable of further movement and replication. So if you're new to transposable elements, I hope that this blows your mind that, up to 2/3 of our genome is DNA that either currently is or once was mobile. Most of it is no longer mobile, but there are still active transposable elements in the human genome, and there's active transposable elements in almost every genome that's ever been looked at. So we have been interested in measuring the mutation rates of transposable elements within a lineage, but we've also wanted to look at this horizontal transfer phenomenon that I started with, the Peter Parker example, where we look to see if there's evidence of pieces of DNA from one lineage making it into another lineage. And this kind of work has been really facilitated by living in the whole genome sequencing era, and the fact that we can sequence whole genomes of many, many different organisms. We don't have to do mutation accumulation experiments that take years and hundreds of thousands of dollars. We can do this type of research simply riding on the coattails of publicly available data that people have sequenced for many reasons that I might never even understand, but is all publicly available now. So we can search for highly similar sequence between, in this example, Tasmanian devils and any other lineage. And if we see DNA with very, very, very high identity, meaning almost exactly the same between one species and another, that region is a candidate region that has horizontally transferred between those two lineages. And we do a lot of quantification of horizontal transfer events among taxa. So what are the big questions in our lab moving forward? We want to know what causes that variation in mutation rates that I showed you in Daphnia magna in particular. That's the system we'll continue to ask those questions in, but we're hoping that if we can start to parse what explains that variation, that it might be something that's we can generalize beyond Daphnia magna to other species. We're really interested in the phenotypic effects of spontaneous mutations. So that is not disease-causing variants, but how variation affects complex traits like height, weight, growth rates, fecundity, et cetera. We're very interested in partnering our estimates of germline mutation rates with estimates of somatic mutation rates. Somatic mutation rates are much, much more technically difficult to measure, but we are now entering the next phase of the whole genome era, which is to say we're entering the single cell whole genome sequencing era. And that ability to sequence the entire genome of a specific cell should let us start to parse how many mistakes and how much damage is happening that's heritable in the genome and how much is happening in cells of different cell types in different locations in the body. And that's another set of questions that have a lot of the same motives as the ones I shared with you at the beginning of the talk today, but that also have other tentacles that extend even further. We still work on horizontal transfer, still looking for cases of horizontal transfer that are interesting, but also trying to shift to what causes horizontal transfer. What is the mechanism that can explain the shuttling of DNA from one species to another? That said, I wanted to do a brief coda before we open it up for questions. We found cases of horizontal transfer of transposable elements among many species. This was 10 years ago, before I started at Reed, was the first case of widespread horizontal transfer of DNA among eukaryotes. So this was a big surprise when we discovered it, because people think horizontal transfer happens all the time in bacteria. Bacteria just have their outer cellular membrane. They have no nuclear membrane. There's very little barrier to horizontal transfer. And in fact, bacteria have sex via horizontal transfer, and that's quite a regular part of the life cycle, if you will, of bacteria. But eukaryotes were not thought to readily exchange DNA that would then be inherited. And so when we started to find that that was happening in dozens of cases, and now there are thousands of cases of horizontal transfer among eukaryotes, this really started to catch people's attention. And part of the reason why we're particularly interested in transposable elements being able to horizontally transfer is that when a transposable element gets from one host genome into a new genome, it doesn't just stay still like regular DNA will. It can then start to jump around in that new genome. And in fact, it is likely to in that new genome where there has not been the coevolution of suppression mechanisms from the host genome fighting that transposable element. So we are very interested in this, and it has led to a bunch of projects in our lab looking at cases of horizontal transfer. And it also led to a citizen science project that we did with collaborators in 5 different countries in East and Central Africa. This happened during my last year-long sabbatical in 2013-14, and the papers have just come out in the last 2 years. This was especially exciting because we were interested in Buciola fusca, this lepidopteran, this moth, because it had been implicated in a horizontal transfer case that we found while mining public databases. But there wasn't a whole genome sequence available for Buciola fusca. So I proposed to Fulbright Foundation to get money to sequence the whole genome. And the argument was twofold. One is that we wanted to understand more about horizontal transfer, but more importantly, that Buciola fusca is the most devastating crop pest in most of Africa, and that there were a lot of reasons that we would have to sequence it in order to understand possible avenues for biocontrol or pest control in order to increase food production. So this side project, I guess is a way of saying it, really took off and yielded a whole genome sequence and publication of the whole genome for Buciola fusca, the whole transcriptome for Buciola fusca as well. And it was done with about 75 co-authors, partners from countries that have a real stakehold in understanding the genomics of Buciola fusca, not just us with our curiosity about horizontal transfer, which might also be related to biocontrol ultimately, but it was a really cool opportunity to make those questions meet. And this is just to make sure that I didn't, on the first slide, I gave credit to the folks that did a lot of that Daphnia work. Here, I want to make sure that I credit the folks that were part of that consortium. So also important to acknowledge the funding. Reed has been a huge supporter of my research program with my startup package and subsequent support for sabbaticals and summer scholarship. I've also been lucky enough to get support from NSF and NIH for this work, and from the MJ Murdoch Charitable Trust, which has done a lot to support undergraduate research in the Pacific Northwest. So I want to, in addition to all the thinking and labor that has gone into this, it did require dollars and cents. So thank you to all of those folks for their help. With that, I want to open it up for questions and say thank you to all of you. There was a question about the horizontal transfer that is a technical question. How does it happen? The answer is we do not know. There are a bunch of possibilities. It could be a viral vector. It could be uptake. Someone asked a question earlier about, is crossover a mutagenic phenomenon? You can imagine when crossover happens and there's a double-stranded break between two sisters, and that break gets repaired. One of the ways that it could be repaired is if a transposable element is used to repair that region. It could also be a piece of foreign DNA that gets incorporated when that's repaired. So all of these mechanisms are highly intertwined in ways that we don't yet understand, and we don't study the mechanism at the molecular level in my lab. We kind of look for the signs of what's happening in between which taxa, and then try to share that discovery with other people who are then going to go in and see if they can put organisms together and make things happen at the molecular level. So we're looking for broad patterns that might suggest how and why, but we're usually not able to test how and why. We're just at the front lines trying to see those patterns. But that's it. That's all I have. I'm happy to take any questions that people have about that. I hope I didn't go too long.
Yeah, thank you, Sarah.
Oh yeah, the kissing bug example. So yeah, one of the cases of horizontal transfer that we found was between about 11 different species. I'll stop sharing my screen unless we need to go back to that. 11 different species of eukaryotes, one of which was a species called Rhodnius prolixus in Latin. It's called the kissing bug, or the assassin bug is the common name. And this is a bug that feeds primarily on blood, and its preferred host is the opossum, but it will take blood meals from many other vertebrates, and it will take blood meals from humans too. And when we looked, we found several different types of transposable element that were nearly identical in the genome of Rhodnius prolixus and many of the lineages that it feeds upon. So that is a great example. What's the mechanism there? Like at a fine scale, we still don't know, but this is a blood-sucking bug that has a anticoagulant saliva that's often deposited in the host when it takes a blood meal. So there's exchange of fluid both ways. Hard to know who's the donor and the recipient when you have fluid exchange of blood going to the bug and saliva going to the host. But that could be an explanation of how that DNA was shared. Of course, we found the same sequence in some lineages that those assassin bugs have never been known to take a blood meal from. So it's more of a smoking gun than a surefire explanation for the mechanism. But based on how many cases of horizontal transfer have been reported in literature since 2010, when we got that big paper in Nature with that case, and that is thousands and thousands of cases, there are probably multiple mechanisms. It's probably not just one. Yeah, and the reason why Rhodnius prolixus was sequenced was because people were interested in Chagas disease. It was not because they were interested in horizontal transfer, but the data were there in the database free, and I don't have a lot to do on the weekends, or at least I didn't when I was a postdoc. And so scouring the database for evidence of this exchange was like a cottage industry for me for many years. Now I usually spend time with my kid. But you can glean a lot from the shared data in those databases. You do have to be super skeptical because of mislabeling, contamination. There's no peer review of what is deposited in the database. It's just a free-for-all. So I can sequence a salmon and I could upload it and say it was David Griffith's genome, and no one's going to stop me from doing that. And so then when I go to search for it and I find David Griffiths has had a horizontal transfer case with the Tasmanian devil, it was a salmon, so you have to really double check and validate any of the events that you find in that public domain, but it is a really good way to cheaply and quickly scan tons and tons of genomes. Yeah.
Yep. Are there other questions?
Do we have in-house sequencing? No, we do not. We can do the sequencing reactions here for Sanger sequencing, and then we can send them either across the river to OHSU or out to a company where they're run out on a capillary gel, and we'll get the data back the next day. But this is all primarily done with Illumina sequencing, or a combination of Illumina and PacBio sequencing. These are all sequencing platforms that are unfortunately referred to by their commercial names, but they are high throughput sequencing technologies that allow you to sequence much larger volume. And we don't have any of those machines here at Reed. We don't have enough sequencing demand to keep one of those machines busy. So whenever I do whole genome sequencing, I send it out and negotiate prices with places that will take external requests, and that's probably fine for some time. I think there's occasionally been talk of getting a tabletop high throughput sequencing here, but it also requires probably a human to make sure that it stays running. So that's another big expense. If anyone knows how to contact, that's a different topic. What's the significance of the much greater rate of mitochondrial mutation? So in animals, generally, the mitochondrial mutation rate is much higher than the nuclear mutation rate. In plants, the mitochondrial mutation rate is much lower than the nuclear mutation rate. And this is another big puzzle in evolutionary biology, why that is true. And the answer is not known. If base substitution rates are broadly similar between organisms over evolutionary time, aren't all organisms about equally evolved? I don't know we're all equally evolved. Every extant lineage on Earth, assuming life arose one time, is equally evolved. And there's good evidence that life only arose once, though it's not for sure. But we're definitely no more evolved than the bacteria under our fingernails, if that's your question. I had fun this year on the qualifying exam for the juniors. I asked, how do people measure organismal complexity? What are the units? And I got so many different answers, including there are no units. That's not actually something you can measure, which is the answer I favor, but it was fun to hear people's discussion of that idea, because it's certainly when you talk with people in the public, the idea of mutation and evolution almost always is directed towards some notion of complexity or higher progress. People refer to the lower vertebrates, and that language is misleading. Have you compared mutation rates between your sexual reproducing Daphnia and asexual? Yes, we have. The nuclear rates don't differ, but the mitochondrial rates seem to differ between them, and we don't know why. Is the viability of individuals noticeably different depending on the different type of mutation? That's a great question. We do not see so far. I'm still analyzing the data. So the basic question that's being asked is, in our experiment, we have all these lineages that we measured the mutation rate in, we know some of them have the highest mutation rate and some have a lower mutation rate. Do we see a commensurate decline in phenotype in the lineages with the highest mutation rates? And it turns out that was totally the goal when we set up the experiment to be able to answer that question. All of the mutation rates, depending on mutation types, are so all over the map. So the genotype that has the highest base substitution rate does not have the highest transposable element mutation rate. And that genotype doesn't have the highest microsatellite mutation. So because none of the mutation rates co-vary across the types, it's been very difficult. It's basically a very ugly multiple regression to try to see if there's a relationship between mutation and phenotype. And so far, we don't see much. I guess I'll say that. But that's, my sabbatical starts in 4 days. And that's the first paper on the docket to be thinking about. Does CRISPR enable any new experiments that could help me? And there is no CRISPR working in Daphnia yet. It's probably will happen. The trick there is just a technical trick of injecting eggs without destroying them. The thing that I think I would do with CRISPR would be to, if I really wanted to get at mechanism, I would start to individually knock out different DNA repair genes and see how that affected the rate. That's not really probably my quest in life to do that. But I do think CRISPR is fascinating and I love reading about other people using it. I'm not sure that delineating which gene it is that explains high or low rates of DNA repair is going to be in my particular opus. Does your work have implications for dating divergences in populations? Absolutely. That's a major application of knowing mutation rates, although there's good evidence to suggest that mutation rates can change over time. So you have to, we think of them as being kind of a rate that is clock-like, but next level of depth of detail would be to look at how they change over time, which I'm not sure how to do in my lifespan, but would be really interesting to do. What can you say about phenotypic expression of these mutations, since that is necessary to close the loop? Yeah, as I said in the reply, it's the declines in phenotype over the course of the experiment were noticeable, but very small. And the mutation rates were so all over the map that it's not a clear strong negative correlation. What am I doing on my sabbatical? Usually I would have it all planned out by now, but because of the pandemic, it's been really difficult to plan any travel or basically commit to anything anywhere. So starting Friday, I'm going to think about that. I hope that I will be able to, I hope that I will be able to basically resurrect my scientific brain after what feels like a 2-year hiatus, just kind of going on fumes and barely, doing a lot more student support than science mentorship, to be totally honest. And that's fine. That's good. That's necessary. But I'd like to have a creative time. I'm, 20 years of studying Daphnia and I love it, but I want to make sure that I either use this time that I have to think deeply and creatively and pivot to a new chapter, or that I reaffirm my commitment to this line of inquiry. Both of those are actually, I think, on the table for me, and I'm just as excited as you are to find out which one I end up choosing. Most importantly, it's just that there is going to be a sabbatical.
Sarah, I think there was a question a little bit back. Why is the mitochondrial mutation in primates referred to as the, quote, molecular clock?
Oh, you can refer to any mutation rate as a clock if you have good evidence that it is not variable over time. So it's just that wherever you saw that they must have been talking about, it's something that was calibrated with the primate mutation rate, but you could be studying birds on Hawaiian Islands and you would use a bird rate, and it probably wouldn't be from the species of bird that you were actually studying. It would probably be from chicken or maybe zebra finch. some other species of bird, and it would be assumed that it's going to be the same for all birds. And what I've told you over the last hour is that that is not a safe assumption. But people calibrate molecular clocks with the mutation rate from whatever is nearest in terms of closest lineage, most recent common ancestor, and they just kind of hope for the best in terms of dating the event that they would like to date. Sometimes they'll combine it with geological events or fossil evidence as well in order to try to make the date more accurate. We use molecular clocks often in order to try to date the invasion of genomes by a given family of transposable elements. So we also use molecular clocks, but it's always relative to whatever nearby lineage you have a rate for. Thank you so much for this opportunity and for the fantastic questions. This is really a pleasure. It's a great way to kind of launch my sabbatical, to take stock of what's been going on in the lab for the last 10 years since I got to read, and think about what's going to happen in the lab for the next however many years. It's an exciting time for me, and I really appreciate your obvious attention and care in listening.
Sarah, thank you so much again for joining us. We really appreciate it. Best wishes with your upcoming sabbatical, of course. And for everyone else, thank you for attending. We hope to see many of you in June at reunions on campus. And with that, have a wonderful day. Thanks.
Thank you again for the great questions. Thanks for the invite, Shanti.
Thank you.